Hi, I'm Dr. Carl Good, a retired molecular geneticist. And today I'm going to provide a presentation on why we age, some new life extension techniques, and at the end, something special about women. Fortunately, with the internet, I can keep up to date and find a lot of the new developments, which I'm going to provide here from my browsing history. In the past, when we were hunter-gatherers, we had limited resources. So if everyone lived too long a life, there would be no food for the next generation. So biologically, we got rid of old people so children can have more to eat. What you see here is a list of maximum ages for a number of different types of organisms. For chimpanzees, it's hard to make it up to 60 years, but for humans, we can make it over 100, perhaps 120. So what's the difference? Some people speculate that grandparents were useful. So keeping them alive would help with work at home or help with hunting. But eventually, there was a case where great grandparents were a little too much of a good thing. So we extended life so the grandparents could contribute, but then that was it. The ancient Greek Heraclitus said no man ever steps into the same river twice. And that was because the river is flowing. So if I step in the river today and then I do it tomorrow, it's not the same water, but there isn't an organization of the river. So the river is still there, but it's not made of the same stuff. So what we classify as a river really has to do with the physical organization rather than the water that is its component. Humans can be thought of in the same way. We're continually replacing the components of our bodies. And what you see here is a list of different cell types and their ages. So we're continually replacing all sorts of cells that make us up. And perhaps the longest living ones are bone cells. But that just means the cell is there. Even more important is that we're continually replacing the components of the cell. So the chemicals like cholesterol and proteins and other things are continually being replaced in a cell before it dies. One of the hot new areas in genetics is something called epigenetics, and it just means things that are not part of the DNA sequence, but that control what the DNA does. So I would assume that no one listening to this has an eye on their big toe, but all the genes required to make an eye are in that big toe. So what's going on? Well, what's going on is we have a system that turns on genes when they should be there, let's say to make an eye in a developing embryo, and keeps them off in other tissues where we don't want to develop an eye. And this is called epigenetics. And specifically, we do this by adding methyl chemical groups to the DNA and the proteins, especially the DNA, to control which genes are actually turned on where. And one of the theories of aging that I subscribe to is that as we age, that regulation of what's turned on where starts breaking down. So we have some things turned on where they shouldn't be, and others turned off when they should be on. So that aging is really a lack of control. As we said before, we're continually rejuvenating, but As we age, that rejuvenation process decreases because of lack of control. There are about 100 children in the world with a genetic disorder called progeria, where they look like they're aged when they're only 10 to 12 years old. A couple who were MDs had a child with this disease in the Boston area and created the Progeria Research Foundation, which has been very effective in studying this disease and providing therapies. In the past, a child with this disease might only make it to 10 or 12 years old, where now there are some that are in their mid-20s due to therapies that were developed by the Progeria Research Foundation. So there is hope that through some of these studies, we are going to learn even more about the aging process. Next, we're going to talk about some of the life extension techniques that are being investigated in research organizations, some funded by the National Institutes of Health, that will help us understand how to extend human life. We're going to talk about why you need to feed your mitochondria, and also it might be good to get rid of bad molecules in blood plasma. Multicellular organisms, like us, are really two types of cells. We have the basic cell that does most of the work, but in each cell, there are smaller cells called mitochondria, perhaps around 500 or 600 per cell. And these can do amazing things. If you're a yeast and you eat a molecule of glucose, you can get 3 units of energy out of that glucose. However, with our system and the mitochondria, we can get 32 units of energy out of that same glucose molecule by taking it apart very carefully. Now, for the mitochondria to work, it needs a substrate, adenosine diphosphate, ADP, which it converts using energy from the glucose into ATP, adenosine triphosphate, which is a general energy molecule, like some others in the cell, that are used to build proteins and carry out chemical reactions. They provide the energy for these systems. However, if we're short on ADP, then the mitochondria will not function as well. And it seems that as we age, we have less ADP than we could need. It's sort of like having a production line and the engines are in short supply. Well, that slows down the entire production line. So we need the substrate or the components of this system to be in high enough concentration. And there are new substrates that can be obtained commercially that will increase the amount of ADP in the cells of older people. Experiments were performed some years ago where 2 mice were connected via their blood supplies, an old mouse and a young mouse. What happened was the old mouse appeared to get younger and the young mouse appeared to get older. So that begs the question as to, was there something good in the young mouse's blood that helped the old mouse? Or was there something bad in the old mouse's blood that hurt the young mouse, but the young mouse's blood helped the old mouse? Well, it turned out there was something bad in the plasma of the old mouse. Well, the question is there some other way than hooking yourself to a young person to get this same effect? And it turns out there is. There's a process that's done quite a bit around the world called plasmapheresis. What happens is it's like donating blood, but a machine separates out different components of the blood and gives most of it back to you. This is used for some autoimmune disease and other situations where the blood needs to be cleansed. Well, if you're going to remove the plasma, but save the cells and put them back into the patient, you need to replace the plasma and primarily albumin, which is a major protein component of your plasma. So in these cases, they'll take human albumin and replace that that's been removed by the plasmapheresis process. There's an interesting thing that I ran into in reviewing material for this talk, was that they've taken human albumin genes and put them in rice. And so you can have rice that now contains human albumin, and you can purify the human albumin out, and then with appropriate salts, give it back to a human. And people will say, well, that's genetic engineering and it's scary, but in reality, albumin produced in rice is better, because if you take it from humans, there could be viruses and other materials in the human albumin that are going to be put into the individual going under the process. But there aren't any things like that in rice. So in reality, it would be better to use human albumin from rice to supplement the albumin that has been taken out with the bad things that are causing us to age. So we want to cleanse our blood, take out the bad stuff, and replace the human albumin with albumin from rice. There is a lot of excitement today about a molecule called rapamycin. It was discovered on the island of Rapa Nui, which we know by its common name, Easter Island. It was thought that this material from a fungus might be antibacterial, anti-cancer, and none of that really worked out all that well. However, it was determined that this molecule affects a central component of our metabolism called mTOR. And TOR means target of rapamycin. And by turning off this mTOR process, it seems as if we go into a repair stage where we replace things in our cells that are too old or ineffective. So rapamycin is getting a lot of study today, and I believe there are even trials for its use in humans to use it as an anti-aging pharmaceutical. The slide you see here is a summary of many of the materials that people believe have an effect on aging. Some of them I've discussed in this talk, and others I haven't. But the whole essence of it is that we can continually rejuvenate ourselves using these types of materials. And as we age, that rejuvenation slows down. So there is no functional limit to how old we can be. I am just adding this last slide as somewhat of a curiosity. As most people know, women have two X chromosomes and men have an X and a Y. So in men, their X chromosome comes from their mother in all their cells, where a woman has a backup plan. So if there's a problem with mom's X chromosome, perhaps dad's can take over. And I'm going to add another little curiosity, is that mammals are very dosage sensitive. There's a disease called Down syndrome where just a small part of one of the chromosomes is there in 3 copies, and that causes the syndrome. So what about the X chromosome? It's very large and would have a very significant imbalance in dosage. So what women do at about the 64 cell stage of their development is to inactivate one of their X chromosomes in each of the 64 cells, and this is done randomly. So part of their tissue has mom's active and the other part has dad's. So even though there's only one active per cell, they benefit from having both X chromosomes. And you can see this in the cat, where they have a color gene on the X chromosome. And you can see in the picture below, the cat has patches of different color because in one patch, dad's X chromosome is active, and another patch, mom's is, and we can see this because of the color difference. So in summary, feed your mitochondria, get rid of bad molecules in blood plasma, consider ADP and rapamycin supplements, live a healthy lifestyle, and remember, you don't have to die.